Today's discussion, we're going to discuss about packaging of DNA. Let's try to understand what does it mean, okay? Now, you know that inside the cell, there is a nucleus. Now, in the nucleus, you found your chromosome, we found our chromosome, we found the DNA, okay, and we found some proteins. So, just think that the cell is so minute, okay, and the nucleus is more minute again, okay. So, in that minute structure, small minute structure, how those DNA proteins are arranged and kept. So, in order to understand this, we have to understand the packaging of DNA, how DNA are packaged inside the nucleus of the cell. So, as you know that prokaryotes or eukaryotes, the prokaryotes doesn't have a well-defined nucleus, okay, whereas the eukaryotes, they have well-defined nucleus. We'll discuss later on the detailed difference between prokaryotes and eukaryotes based on their packaging of DNA. The length of the human DNA, okay, length of the DNA in human genome is 2.2 meter. So, just say that one genome is nothing but haploid set of chromosomes, okay, and haploid set of chromosomes. One thing you have to know that DNA is always negative charged. Negatively charged. Okay. There was a scientist called as Woodcock. W-D-O-C-K. Woodcock. Okay. He observed in a chromatin fiber, he observed a thickening like this. He observed a thickening, okay, in a chromatin fiber. What did he observe? He observed a thickening in a chromatin fiber, and he did not know what is that, okay. So there was another scientist, okay, who is known as Oded, who gave that term as nucleosome. He said that it is a nucleosome, okay. So we're going to discuss the structure of what this nucleosome is all about. This nucleosome is made up of histone molecule, okay, and DNA. Now, histones are usually positively charged and they contain two rich basic amino acids, okay. It is lysine plus arginine. So, this is very important to know in your NEET examinations. These questions was asked, okay. So, this histone is a positively charged. It has a basic amino acid, lysine and arginine, whereas DNA is a negatively charged. So, the component of nucleosome is this, okay. Now, what is nucleosome? When this histone wrapped with DNA, okay, when this histone wrapped with DNA, they are said to be nucleosome. I'll show you the structure. Just say that histone. Histones are said to be octamer. When I say octamer, it is 8. So 1, 2, 3, 4. Okay. Now, this is H2A, and there will be another H2A on the back side. Okay. This is said to be dimer. Okay. Next is H2B. And you can say that another H2B is on the back side. Okay. Likewise, this is H3, this is H4. Okay. Likewise, there will be H3 and another H4. So you get dimer molecules, H2A, H2B, H3, H4. Okay, so it will be multiplied by 2, you get 4, you get 8. So octamer means 8. Okay. So this is about the histone molecule. Now the DNA is going to wrap around it. Okay. So I'll just show it here. So this is your histone. I'm making it ball. Okay, just one, showing one molecule, but it is octamer. Now here the DNA is going to wrap. Black one I am mentioning by red. Now after, just set that another histone molecule here. This is another histone molecule and this is wrap around it, okay, like this. So this is nucleosome, one nucleosome, this is another nucleosome, just say, okay. So this is our, the black one is our DNA, okay, which is wrapped around. Okay, so this structure are said to be nucleosome, but this ball-like structure which I made, which is octamer, okay, if it comes so close together, so they may collide. Okay, so there is a clamp like this, clamp, and which we call H1 histone. Okay, here also you will get a clamp which is known as H1. Okay, but this is not involved in the process of inheritance, so it was not included. Okay, so only the nucleosome means histone and DNA will be involved in the inheritance process, but H1 is not involved. So when we say nucleosome, we include only H2A, H2B, H3 and H4 molecule. Now this, from one histone, from one nucleosome to another nucleosome, this is a nitrogenous base sequence. This is usually called as linkers. And these sequences of nitrogenous base is about 60 base pair. Okay. Now the nitrogenous base or the DNA, okay, which is wrapped around the histone molecule is 146 base pair. Normally in other books you will find 200 base pair. Okay, so this is the structure of your nucleosome. Now this nucleosome, when they pack, when they are so closely packed, means densely packed, we call as heterochromatin. Just say that this is our chromosome and our DNA, and then here is our histone molecule. I'm making in a dark band. Okay. So these dark bands are said to be heterochromatin. And this is inactive form, okay? Inactive form. Now, I'm making a light stand-like structure. This is also nucleosome, but this one is light stand, okay? This is dark stand, which is inactive form. It does not involve in the process of transcription. Okay, this is in a light form. This is called as euchromatin. Now this will be involved in the process of transcription. Okay, it means that this is light stained and active form. So this is the packaging that we are discussing. It is in the case of eukaryotes. Okay, you will not find nucleosome or histone molecules in the case of the prokaryotes, because in prokaryotes, just say that this is an E. coli cells. Okay, it doesn't have any cytoplasm, it doesn't have any nucleus, no well-defined nucleus. So their genetic material, just say that this is their DNA. Okay, genetic material. They don't have any histone molecule. Okay, so this, but their DNA also wrapped with some proteins. Okay, they also wrapped with some proteins, and we call that as nucleus. In case of prokaryotes. So this is how the DNA is packaged in eukaryotes, okay? And for prokaryotes, we say simply as nucleus. Same thing, DNA is also negative charge here also, okay? And this protein is also positive charged, okay? We don't find complex structure in the case of prokaryotes. Now, in the next class, we are going to discuss about the process of transcription, okay? Since we discussed on the replication, it was DNA to DNA, then DNA to RNA, then RNA to protein, okay? So, in the next topic, we are going to discuss some of the important processes that is required, okay, for the process of transcription. Thank you.